wanted to know, like Christian.
A technology of developing, for example, plants from other materials other than the normal materials. We can use cells, we can use callus, we can use plant tissue, we can use a part of a leaf, we can use a part of the fruit, other means other than the normal means we know, the seeds. Why is that important? We have problems like diseases, right? When you use seeds, you will have materials which are diseased. But now, if we extract materials like meristems, we are in position to produce clean materials. Also, when we are producing seeds, for example, in bananas, you have to wait for 24 months for you to get the suckers and unplug. But with this technology, we can just get a sucker, cut it in 2 pieces, and within a period of 3 months, we are in position to have thousands of planting materials.
Ghent University, under the leadership of Galnick and the system, has supported the production of a number of PhDs up to 25 trained under the Virio S capacity building program and other masters as well. The second thing is that now we want to have high-spec laboratories that these PhD holders can work in and also compete to entrench their aspirations with partners in Belgium. And as a result, we have received the Biotechnology Laboratory, which is going to support in the tissue culture scientific work. What does this mean? We want to do science to improve productivity of our plant species, which must be resistant to climatic testing and challenges, but at the same time, improve production and yields within the farming communities. That means we make more money because the varieties are competitive. That means we transform the economy. So,
regarding the lab, it is at world-class level. I mean, in terms of the technology, the concept as such, it is standard and in line with modern, up-to-date technology. It will allow the university to train people in a very specific technology, namely tissue culture technology, biotechnology, and in that way it will offer the opportunity to develop new crops, new and adapted crops, adapted to
the biotechnology lab that specifically is going to micropropagate crops. Particularly we start with coffee and banana, and progressively we shall move on to other relevant crops for the economy of Uganda. It will not only do research, but also we are going to use it as a practical training center for our students who are doing agriculture and related biological sciences.
So we will adhere to very strict SOPs, but the facility here is extraordinary because in East Africa this is the only facility of this class, which is really exciting because it will allow us to use this facility for a lot of applications that usually are hard to achieve in a regular laboratory setting.